So today, the topic will be single-cell splicing QTL links cell type-specific splicing to complex diseases. Thank you for your time in joining this seminar. I started my career as a statistical geneticist, and at the beginning, actually, this is before I started doing statistical genetics research. The first successful GWAS was conducted by the WTCCC consortium back in 2007. So as you can see, at the first completion of the first successful GWAS, the number of signals across the genomes are quite sparse. And you can clearly count the number of signals. But then in 2018, after about 10 years of development, the number of signals has expanded significantly. So in fact, there's a funny story behind this because the GWAS catalog makes these figures every quarter. But then this is the last figure on the right that they made because there's just simply no space to insert more GWAS signals, and it's too full across the genome. So we know that GWAS loci are quite important, but they're very hard to study because of they're mostly situated in non-coding regions. So we know that 90% of GWAS loci are in non-coding regions, whereas 10% of them are in coding regions. So even though we think that GWAS is the core of modern human genetics and it's instrumental in finding these disease-associated signals, in order to translate GWAS loci into actionable target genes, there's a much larger body of work, which is functional genomics. So I spent about 6 years to work on this project called the Genotype Tissue Expression Project, GTEX. So I'm showing here the latest published version, which is version 8, and it has about 338 donors across 49 tissues. So as you can see, this includes the lung, the brain, the heart, most of the important organs in the human body. So the results from this paper is multifactorial, but there's one thing that I would like to highlight. On the figure to the top right corner, on the x-axis is the negative log 10 p-value, right? And on the y-axis is the proportion of SNPs surpassing that p-value. So if we set a nominal threshold of 0.05, 97% of SNPs would pass this threshold, which means that, if we use a nominal threshold, most of the SNPs will pass this threshold. But this threshold is stupid because, no one would actually use this nominal threshold. So if we correct for multiple hypothesis testing, correct for the number of tissues that's tested, 64% of SNPs would be associated with expression of at least one gene, right? So still very high. And if we restrict the proportion of SNPs to be 0.05, then we'll have to use a negative log 10 p-value threshold of 24.84, which is insanely high p-value cutoff. So the take-home message of this figure is that using the GTEX data set, a multi-individual population scale across a large number of tissues, we have enormous power to detect as the association between SNPs and gene expression. But even with this enormous power to detect these associations, initially we thought perhaps we could map more than, perhaps even more close to all the GWAS loci. But in fact, across the 20 traits that we have analyzed here, less than half of GWAS loci actually co-localize with GTEX eQTLs. So some traits performed well. So for example, celiac disease, close to 80% of celiac disease GWAS loci co-localized with GTEX QTLs. But for some other traits, for example, for lupus, less than 20% of lupus loci actually co-localized with GTEX QTLs. So this begs the question of where are the remaining co-localization events? right? So from 2020 to now, the field has moved past GTEx and moved on single-cell QTLs. And we're realizing that there are three key pillars to analyzing or identifying these uncolocalized GWAS loci by bulk sequencing. And the three key pillars is that we need to look at the correct cell types because many of the colocalizations are cell type specific. And we need to look at the correct differentiation stages because some of the QTLs only shows up in certain cell types that are not terminally differentiated. And also we need to look at the correct perturbations. So for example, BMI, comorbidities, or smoking. So sometimes, some QTLs only shows up when there is perturbation. But today, in this talk, I'm here to argue that besides these three key pillars, we'll have to look at also the correct ancestry, as well as the correct molecular phenotypes. So here is a summary of the single-cell QTL papers published from 2020 to now. And so actually, some of the papers are from Australia. And all these papers suffer from two problems, right? So the first one is that all these papers analyze European populations. And the second problem is that all these papers analyze one single molecular phenotype, which is the gene expression, right? So which begs these two questions. What about non-European populations? And what about other phenotypes? So for example, RNA splicing, right? So going after these two goals, we constructed this consortium called the Asian Immune Diversity Atlas. So on the top right here is Sham and Ken Hong. So these are the two key leaders for the Asian Immune Diversity Atlas. So this atlas leverages the unique properties of the Singapore population as well as other countries, including Japan and Korea. So we sampled more than 500 individuals and sampled their PBMCs from their peripheral blood with over 1 million cells. So here is the cell type categorization, right? So some of you would recognize these familiar cell types. There's dendritic cells, there's monocytes, there's natural killer cells, T cells, and B cells. And there's also subcell types, right? Gamma delta T, CD8 cytotoxic T, and helper T cells. So using these cell types, we were able to, actually before that, let me talk about splicing. So as I mentioned before, most of the papers analyzing single-cell QTLs are focused on one phenotype, which is gene expression. This is justifiable because people thought that splicing will be much more challenging than expression. And because the expression matrix is already very sparse, it will be even more challenging to analyze single-cell splicing. And thus, there's a scarcity of work focusing on single-cell splicing. So we thought to test the feasibility of doing single-cell splicing, and here is what we have found, right? So these are different cell types across the PBMC landscape, and across all the cell types, we were able to identify between 1,000 to 2,000, alternatively spliced genes per cell. So this is actually comparable to the number of expressed genes per cell. And I'll talk to you about why we could do this in a second. But compared to the previous state of the art, which is we used the 1K data, we were able to identify about 267 alternatively spliced genes per cell using the 1K data. So using the IDA data, we're able to push the number of alternatively spliced genes detection by as much as 5-fold. And when we were submitting this paper, the reviewers asked us, are you just sequencing more deeply? So here on the right, I'm showing you that that's not the case, because on the x-axis is the number of expressed genes, which is linearly correlated with the sequencing depth, right? So the more you sequence, the more number of genes you detect to a point, right? To a saturation point. On the y-axis is the number of alternatively spliced genes. So as you can see, the number of alternatively spliced genes and the number of expressed genes are linearly correlated. The more expressed genes, the more the alternatively spliced genes. And using 1K, 1K, for every 100 expressed genes, we can detect about 12 alternatively spliced genes, whereas using the IDA data set, we were able to detect about 66, right? So, and I'll show you why this is the case by unskipping one slide. So the key change is actually quite simple. Most single cell data is generated by 3' capture. But for IDA, we used a less commonly used technique, which is the 5' capture. So why does this make a difference? When in normal cells, there is a naturally occurring phenomena called stochastic mRNA cleavage and recapping. We don't take credit for this. This is actually a phenomena that was discovered by ENCODE back in 2009. So what happens is that in your cells, the cells would actually randomly cleave off the 5' UTRs. This is a protective mechanism in the cells because it generates more diverse transcriptomes without affecting the coding region. So in theory, this would increase the diversity of transcriptomes in the cells and make the cells potentially more fit under, for example, negative selection. This phenomena can be leveraged by 5' sequencing because the 5' sequencing could capture transcripts that starts from different 5' ends. So essentially, we go deeper into the 3' end without paying any additional cost, right? So in total, we saw that there is a linear increase in the fraction of exons covered versus the number of reads per gene, right? So if the gene is highly expressed, then it's more likely for there to be exon pinning effect, and then the fraction of bases covered will be higher. So in total, we were able to cover around 81% of bases across all genes, which is much, much higher than we originally thought. Originally, perhaps we thought we can cover like 20 to 30% at the 5' end. So using this expanded coverage of RNA isoforms, we were able to perform a few analyses. The first one is that we can take a look at the differential splicing landscape across populations, right? So here I'm showing you using different colors, the Chinese versus the Indian population, the Chinese versus the Malay population, and then the Indian versus the Malay population. So I just want to emphasize one thing here, right? So the IDA consortium is all healthy individuals. So here we're comparing healthy individuals against healthy individuals. So the question now here is, why would there be any differential splicing between two healthy individuals, right? So we did observe some of the reasons, and I'm showing you here one example, which is allele frequency drives differential splicing. So here, this is the gene SB2, and this is the key isoform, right? This isoform, I'm sorry. So take a look at this isoform first. This isoform uses a canonical 3' splice site, which is AUGAC. And this variant RS1106447 changes the G variant to an A variant, thus disabling this splice site. So the splicing machinery is forced to search for an upstream splice site, which is this one, this GAC splice site. So we observe that there is a gradient of this variant RS110 across the Chinese population, the Malay population, and the Indian population. So the Chinese population has the highest allele frequency, which means the highest level of disruption. So you see the Chinese population has the lowest usage of this splice site. And for Indians, because they have the lowest disruption, this splice is most commonly used. And you see that the Malay individuals are somewhere in the middle. So this example shows one of the possible explanations for why there is differential splicing across populations that are completely normal and healthy. And one thing I should note is that this variant, this isoform variant, is actually not annotated by gene code. But the black ones are annotated. So this shows the importance of using ancestry-specific transcriptomic data, or otherwise we risk losing these novel ancestry-specific isoforms. So besides single-cell splicing, we can also combine the splicing data with the genetic data to map splicing QTLs. So here we have identified about 11,000 cis-SQTLs across 19 of the 22 cell types, and also about 607 trans-S genes across the 19 cell types. So I'm going to show you two stories of why this splicing QTLs matter, right? So in the first story, I'm going to show you a biological pathway that was identified using our data set, right? So here, first of all, we identified two regulatory variants influencing this, the expression of this gene called HNRMPLL or HNRMPLL. So these two variants are acting through different cell types. So the first variant acts through naive cell types, naive CD4 cell type, and the second one acts through memory CD4 cell type. And the reason I think that these two variants are independent is because that they are 36 kb apart. And also once we do fine mapping, their signals are independent from each other. And also their R squared is less than 0.3. So we're fairly confident that these two variants are both regulatory variants, but they're independent from each other and they're acting in two different cell types. We did a further analysis on these two variants. It turns out that not only are they cis-acting for HNRPLL, they're also trans-splice QTLs for this gene called PTPRC. So in the hematology world, is also called CD45. So this CD45 gene is a multi-exonic gene that's quite important for T cell development. So there's two well-known isoforms. One is called the RO isoform, and the other one is called the RA plus isoform. So the RO isoform does not include exons 4, 5, and 6, and the RA isoform includes these three exons. So we found that these two variants are strong splicing QTLs for the RO versus the RA isoform. And in fact, you see that whenever there is a HNRMPLL cis eQTL, there is a CD45 trans SQTL. So their signals are quite correlated with each other, right? So, so this leads us to a further investigation, which is why is there such percolation of effect sizes? So we found that, by previous literature, that HNRMPLL actually expresses this protein that could bind to CD45 exon 4, 5, and 6. And we can actually validate this effect using our single cell data set because we can stratify the cells by whether they express HNRPLL. So if they're not expressing HNRPLL, then the PTPRC splice isoforms will be much longer. Whereas if they expressed this gene, then the PTPRC isoform will be much shorter. I mentioned a couple of slides ago that CD45 is actually a key determinant of T cell differentiation. So exon 4, 5, and 6 encodes an area that can be glycosylated, which acts as a signaling, which triggers a signaling cascade for CD4 cell maturation. So here we further ask the question whether these two variants would influence CD4 T cell proportions. And the answer is yes. And we did see a very strong signal of the cis eQTL effect and central memory CD4 T cell proportions. So to summarize, we have identified two cell type specific variants that both influence the expression of a RNA binding protein called HNRMP-double-L. And this RNA binding protein would translate, RNA binding gene would translate into this protein, which binds to CD45 exon 4, 5, and 6, which influences its splicing patterns. And such change in splicing patterns would then influence T cell development and influence T cell proportions. So that's story number one. The next story, I would like to connect splicing QTLs to diseases. So here we performed colocalization analysis and we identified dozens of putative causal genes. So here I'm showing you a subset of 30 genes. And for the interest of time, I'm just going to highlight one of the genes, which is TCHP. So this gene acts in multiple cell types, including monocytes, natural killer cells, and T cells. And variations of the TCHP gene would modulate the risk of Graves' disease, right? So this purple color right here. So here, the first thing I want to highlight is that this GWAS loci for Graves' disease is mediated by splicing QTL, but not by expression QTL. And the reason I say that is because we can see that for every cell type where there's junction usage of a specific junction, which I'll show you later, there will be a strong splicing QTL effect. And whenever there's a splicing QTL effect, there will be an expression QTL effect. And this is by nonsense mediated decay, which I'll show you later. And the reason why we don't think that is the other way is because there's no correlation between gene expression and eQTLs. So you see that the pattern between gene expression is so this is ranked by the eQTL strength, and the pattern of gene expression is random, right? So there's no visual correlation between gene expression and eQTL strength. So the second point I want to show is that this splicing effect is genetic, is cell type specific. So here is Graves' disease GWAS, and we see that the same lead variant RS74416-240 has a regulatory effect in CD14 monocytes, CD16 monocytes, natural killer cells, T cells, but this effect disappears in B cells and also all these 12 cell types, right? So it's actually quite important that we look at this signal in a cell type specific fashion, because if we look at, for example, GTACs, which used whole blood, the minority of cell types would have this QTL, but the majority of cell types would not have this QTL, and thus it's unlikely that we can find this colocalization. The next thing we did is that we can validate this the genetic effect of this variant RS744. So here I want to highlight two of our wonderful collaborators, which is Donald Sim and Xavier Roca. They're expert in the splicing field, and they helped us to perform this mini gene test. So here, what they did, before I jump into that, I would like to highlight that this RN744 is an Asian-specific variant, right? So here we show that the allele frequency of this variant in the Japanese population, in the Chinese population, in the Korean population. As you can see, in East Asian populations, the allele frequency is quite high. But this allele frequency decreases in the Malay population and then disappears in the Indian population, right? So we can also validate this effect using 1,000 genomes, where we found that there is sufficient allele frequency for this variant in the East Asian population, whereas this variant disappeared in European, Native American, African, and South Asian populations. Well, I should justify that it's not to say that European populations, Native American populations, African populations, and South Asian populations are not important. It's just because that we're here using a Biobank Japan GWAS, and thus we're biased towards East Asian populations here. And so we can actually clone out this TCHP exon 4 into a mini gene vector and is surrounding 200 BP upstream and downstream. So we can construct two identical mini gene construct differing only at one base, which is this RS744. And then eventually we can test this by injecting, transfecting this mini gene into 293 cell lines. And then we can identify the different splicing isoforms using a gel electrophoresis. So here you see that the G allele, which is the protective allele, gives us the normal splicing isoform, right? So it's the left exon followed by the middle exon followed by the right exon. Whereas if we change the risk allele, the non-risk allele to a risk allele, then we observe a much larger band, which corresponds to intron retention. So I just want to quickly summarize the findings here. So the first finding is that ancestry-biased alternative splicing is widespread, right? And sometimes they can be explained by allele frequency differences. And this is part of the reason that using the correct population is quite important. And the second point is that trans-splicing effects can sometimes be mediated by cis eQTL effect on RNA binding proteins. So here I showed you one example of two independent SNPs regulating the same gene, HNRMLL, which then trans-regulates this gene CD45, and that changes cell type proportions of T cells. And last but not least, we showed that colocalization can identify cell type specific, can explain cell type specific GWAS signals. And TCHP is one of the example, which is a putative causal gene for Graves' disease. And its risk allele only appears in East Asian populations. And if we had used, for example, Malay population-specific GWAS, Indian population-specific GWAS, I believe we would have found, GWAS colocalizations for these other minority populations. So it's therefore very, very important to look beyond, European populations and into these minority populations. And I just want to quickly look ahead of what's next, right? So here I'm highlighting our work, which is the Asian Immune Diversity Atlas, but it would not be fair to miss other people's work. So there is also the Chinese Immune Multiomics Atlas, which is currently under review. It has 5 million cells. And also there is the Cardinal UK Biobank project, which sampled across 5,000 populations with 10 million cells. And also there is the 10K project, and their preprint went online last week. The phase 1 preprint, and the whole project aims to generate 50 million cells. So we have the data now, but what should we do with the data is the key question. So I think there's 3 pillars of work we need to do next, right? So most groups are sampling PBMCs, which for good reasons, right? Because PBMCs are easy to sample, but we need to look at further diverse tissues, right? So we need to look at, for example, the brain, the artery, the heart, the muscle, the adipose. So for example, our friend Do Yunong here in Indiana is doing amazing work on the brain single cell QTLs. And also we need to look at diverse disease states, right? So one of the flagship projects is called the Single Cell Atlas of Immune Diseases, right? From our Korean colleagues. And also we need to integrate across many data sets, across many different batches and different modalities. So one of my postdocs, Xu Chang, is working very closely in this area. And last but not least, we need to do machine learning modeling, and not only classifiers, but also large language models and even autonomous agents, right? So here I'm showing one example, which is a cell language model. And this is part of the Chinese immune multi-omics atlas paper that's currently under review. So I like to highlight many, many wonderful collaborators, and especially these 7 individuals. So the top row are the 3 co-first authors of the paper. And then the bottom row are the 4 data champions for this cohort. So Ken Hong, Sham, Jay, and Won Yang. And thank you very much. If you would like to know more about my group, you can scan the QR code on the top left corner. And also feel free to reach out to me using the email on the top right.